Okay, so welcome to this video. In this video, what we're going to talk about is the CRISPR-Cas9 system. Okay, so what we're going to first start off by discussing is how this system works in bacteria. Okay, so where we found this system firstly, which is we found it in bacteria and also archaea, which are also single-celled organisms that are very similar to bacteria. Okay, and in those bacteria, basically it acts as an adaptive immune system against viral infections. Okay, right. So the CRISPR-Cas9 system. So we're going to start off by discussing this system within bacteria, where it functions as an immune system. Okay, and specifically, I think the CRISPR-Cas9 system was found in streptococcus pyogenes bacteria. Okay, I think the actual enzyme differs depending on which species of bacteria or archaea you actually look at. So I think this was specifically found in streptococcus pyogenes. Okay, but loads of different bacteria have an analogous system. Okay, right. Then what we'll talk about is the application of the CRISPR-Cas9 system to genetic engineering, and we'll specifically look at how we can make a genetic knockout, for instance, a knockout mouse, using the CRISPR-Cas9 system. Okay, right. So then, we'll start off with the use of this as an adaptive immune system, or something similar to our adaptive immune system within bacteria. Okay, so bacteria hugely outnumber human cells on this planet. The number of bacteria cells compared to human cells is enormous, basically. Most of the biomass on this planet is made up by bacteria. Okay, so there are many viruses which infect bacteria rather than human cells. Okay, and viruses which specifically target bacteria are known as bacteriophages. Okay, now bacteriophages have a rather cool structure. Okay, they're the ones that kind of look like sort of spacecraft structures. Okay, so they have a sort of hub here, and then they have these sort of processes coming down like so. Okay, and what they can do is they can bind onto a bacterial cell and inject in their DNA. Okay, so let's say we're working with a bacteriophage which has a double-stranded DNA genome. Okay, and what's going to happen is it's going to come up to our bacterial cell here, and it can implant its double-stranded DNA into the bacterial cell. So I'll put the double-stranded DNA here in red. Okay, so here's our double-stranded piece of DNA, and this virus can implant that double-stranded DNA into the cell, and then the double-stranded DNA can then basically take over. Okay, it takes over the cell, it hijacks the cell's machinery, and it uses the cell's machinery to make proteins that are viral proteins, basically. Okay, and it will manufacture new virion particles, new bacteriophages, which will then leave the cell and go off and infect other cells. And most likely they'll kill the cell in the process of using it in this way. Okay, right. So now the CRISPR-Cas9 system is a system which bacteria have, specifically streptococcus pyogenes has, but you find an analogous system in many different bacteria and archaea species. Okay, to protect them from this. Okay, so if a bacterial cell happens to manage to survive the viral infection, that's where we start off with. That's what this entire thing starts off with. It starts off with a cell surviving. Okay, so this is quite a rare event. Most bacterial cells are going to die when they're infected by this virus. But we need one to survive, okay? And if it happens to survive the viral infection, it's now going to make all of its descendants immune to this virus using the CRISPR-Cas9 system. So what's it going to do then? Well, let's draw the viral genome here firstly. Okay, so here is the double-stranded piece of DNA of the viral genome here. And this is inside this cell that's going to survive. Now, basically what happens is that this cell will chop up the viral genome and take little fragments of it and implant those fragments into its own genome. Okay, so let's say we're going to take this little fragment here. Okay, so here is a fragment of the viral genome, and basically this fragment is going to end up being implanted into the bacterial genome. Okay, and this is going to serve to help the bacteria next time that this same virus infects it. Okay, so what's going to happen is this is going to be put into a portion of the bacterial cell's genome known as a CRISPR Locus. Okay, so let me tell you now about the CRISPR Locus. So now we're drawing the bacteria's genome. Okay, so this is the long piece of DNA that you'll have within the bacterial cell here. Okay, so here is our bacterial genome here, and this isn't a plasmid, this is the main piece of DNA of the bacterial genome. Okay, and note that bacterial cells do not have nuclei, so the genome is just within the cell. Okay, right. Now, somewhere on our large chromosome, you will have a locus known as a CRISPR locus. Okay, now let me explain what a CRISPR locus is. Okay, I'll firstly just highlight it up in blue here. Okay, well, a CRISPR locus, firstly, what does CRISPR actually stand for? Well, CRISPR stands for clustered, that's the C. Okay, then the RI is for regularly interspersed. Okay, so it's a long name. The clustered regularly interspersed. Okay, and then the S and the P is for short palindromic, and then the R is for repeats. Okay, so this is the clustered regularly interspersed short palindromic, whoops, not like that, like this, palindromic repeat locus. Okay, right, so it contains a lot of repeats basically. Okay, and I'll explain the significance of these repeats in a moment. So that's what CRISPR stands for. Clustered is the C here, R is the regularly, okay, or in fact I'll have RI as the regularly interspersed, okay, SP is for short palindromic, okay, and then the R here is for repeat. So this is the clustered regularly interspersed short palindromic repeat locus. Okay, right, so let's now pull out the CRISPR locus and draw it in a little bit more detail. So basically, the reason it's called the clustered regularly interspersed short palindromic repeat locus is that it has a number of sequences which are repeated multiple times. So let me put these repeats in. So we'll show these repeats in purple. So here is a section of the CRISPR locus, which is a sequence of organic bases, and basically we are going to repeat this identical sequence of organic bases multiple times in the CRISPR locus, okay? So you'll have it repeated hundreds and hundreds of times, okay? And this little sequence here in purple that is repeated loads and loads of times with these gaps in between the repeats, this is known as the CRISPR repeat, okay? So this is the structure that we actually repeat multiple times within the CRISPR locus. Now, firstly, before we go any further, let's talk about the fact that the DNA strands will have a polarity. Okay, they'll have an orientation, and let's put this orientation on this picture. Okay, so we'll say that this top strand here will have its 3' end at this side and its 5' end at this side, and we'll say this bottom strand will have its 3' end at this side and its 5' end at that side. Well, it has to once we've decided on the polarity of the top one, because remember, DNA strands are always anti-parallel to one another. Okay, right, so that means that in this CRISPR locus we've also got a 3' end here, a 5' end here, 5' end here, and a 3' end here. Okay, just having taken that out from here. So this portion, I'll just try and show it smaller in here. Okay, so here are the CRISPR repeats that we've put in so far, but you'll have far more of them than what I've shown here. Okay, right. Now, basically, what happens is when a cell survives a viral infection, okay, what it's going to do is take little fragments of the viral genome, such as this fragment here, and it's going to add them into these spaces between the cluster repeats, the CRISPR repeats, okay? So, each of these gaps here will have a little fragment from a viral genome that this bacterial cell has been previously infected with. Okay, so we'll colour this one here in blue. So this is a fragment from a viral genome that we have previously been infected with. Okay, so this is a fragment from a viral genome. Okay, right. Now, you are going to gradually build up your repertoire of fragments from viral genomes. Okay, so here is one fragment of a viral genome, but as you get infected by more and more viruses and survive those infections, you'll gradually build up your repertoire here. So we might have another one here. Okay.